Alright guys, welcome to our last presentation. We are going to get through this chapter even if it kills us. I'll now introduce you to our final subject, protein structure, which I'll begin by using a brief metaphor. As you can well imagine, if we were trying to describe the Earth's geographic makeup, we might begin by dividing it into two hemispheres. We could then divide each of those hemispheres into continents, and then divide up each of those continents into countries, and then each of those countries into provinces or states, and then each of those states into counties, and so on. To put it another way, when we look at a map of the globe from a satellite's perspective, that is, a very zoomed out point of view, our view is very large. The amount or the scope of geography that we see is very broad, but the amount of detail that we see is quite small. As we zoom in to hemisphere and then country and then state or province and then county, etc., our view or the amount that we're seeing decreases, but the detail increases. I want you to keep this analogy in your minds as I introduce you to this topic. You see, we chemists like to describe protein structure also at different levels. We use 4 levels, in fact: primary structure, secondary structure, tertiary structure, and quaternary structure. I'll now describe what each of these terms means. A protein's primary structure is the most detailed or zoomed in way of describing it. Simply put, the primary structure of a protein is its amino acid sequence. Now, proteins often contain hundreds or even thousands of amino acids, so determining that sequence can sometimes be a challenging task. This task is accomplished more or less by chemically tearing the proteins apart, identifying the resulting fragments' structures individually, and then figuring out, almost detective-like, how those individual fragments went together in the original protein. I'll now introduce you to a few different ways that we tear proteins apart into individual fragments. The first thing that we do is we cleave or tear apart the disulfide bridges. So you might remember me talking in our previous presentation about how cysteine amino acids and peptides can bind with each other to form disulfide bridges. We can cleave that by treating our protein initially with a compound called 2-mercaptoethanol, shown here, which happens to smell like diarrhea. Now, I just want you guys to know, I don't really care if you know the sequence. I just want you to understand that the first thing that we do when we're trying to tear our protein apart is we get rid of those disulfide bridges. The next step we do is we take or try to figure out the number and kinds of amino acids. This is done by breaking the protein apart into its individual amino acid components by heating the holy crap out of it with 6 molar HCl. So I might have a protein that's composed of hundreds or thousands of amino acids. I boil the snot out of it with aqueous HCl, and what it does is it breaks every single amide or peptide bond in the entire protein, hydrolyzes it to give the individual amino acids all floating around in solution. Once I've done that, then I can figure out what amino acids are present in that protein and in what amounts by taking this mixture of amino acids and putting it in an amino acid analyzer. Now, how that analyzer works is covered in section 23.5. I didn't discuss it, but if you want to, you're welcome to refer to that section. Once again, that analyzer tells us how many amino acids there are of each type and what types there are present in this protein. Now, at this stage, we don't yet know what order the amino acids were in. All we know is which ones there were and in what amounts. To figure out the order that they're in, we have to take another sample of the original protein and start treating it with various reagents that selectively cleave or break apart specific peptide bonds, separating that protein out into individual fragments. These fragments can then be analyzed individually It's much easier to analyze and figure out the sequence of a fragment than it is an entire protein. So we analyze the individual fragments and figure out what their sequences are, and then once we do that, we detective-like go backwards and figure out how each of these fragments went together to constitute the entire original protein. Now, there are numerous agents that we chemists can use to break proteins apart for chemical analysis. One of these is called Edmonds Reagent, which selectively removes a single amino acid from the N-terminus of the protein. Now, we can see that in the overall reaction here. I have a peptide drawn here, which could be dozens or even hundreds of amino acids long. This end, where the nitrogen is, you'll remember, is called the N-terminus. And this end is called the C-terminus. Now, this squiggly line here represents all of the other amino acids in this peptide that I really don't want to draw. This could be, once again, a few dozen. It could be dozens or hundreds of amino acids here. What I've done is I've just drawn the last two amino acids on the N-terminus end of our peptide chain. So when I treat this peptide with Edmonds reagent, what it does is it tears off the last amino acid on the N-terminus. So in other words, it breaks apart this bond right here and leaves me this product 2, which you should notice is exactly the same structurally as my original peptide, except that it's one amino acid shorter. Where did that amino acid go? Well, it went into this product, so it's incorporated into this. This product 1 can be analyzed, and once we figure out what this R side chain is, we can determine the identity of the amino acid that was found here at the N-terminus end of the peptide. So we've now figured out what the identity of the N-terminal amino acid was. What do we do now? Well, what we can do is we can take this product 2 and then treat it with Edmonds reagent and cleave off this amino acid. Identify it. And then we can take the next product and treat it with Edmonds reagent and figure out and cleave off the N-terminal amino acid of it and figure out what that amino acid was, and so forth and so on and so forth until we've sequenced or figured out what the identity is of every single amino acid in this entire peptide in order. Now, as you can probably imagine, doing this Edman degradation work is really impractical and tedious for large peptides of proteins. For large peptides and proteins, we instead use enzymes that are called endopeptidases that selectively break only one type of bond in the middle of the peptide chain somewhere. Now, there are numerous endopeptidases, but there are only two that I'm going to require you guys to know or be familiar with: trypsin and chymotrypsin. So, trypsin selectively cleaves the amide bond on the carbonyl side, or on the right side, of these amino acids, lysine and arginine. Chymotrypsin does the same thing, except it only does it to the right side of phenylalanine, tyrosine, and tryptophan. Once again, I hope you understand what I'm saying. If I have a huge peptide and I treat it with trypsin, what happens is trypsin looks down that peptide. Anywhere it sees lysine or arginine, it just breaks the bond to the right of only those two amino acids, and it doesn't touch any of the others. Chymotrypsin does the same thing, except it does it for phenylalanine, tyrosine, and tryptophan. So are you ready for some questions? Good. Let's take a look at my first lecture problem for the day. I want you to give me the amino acid sequence of the hexapeptide. That is, a peptide containing 6 amino acids that contains arginine, glycine, isoleucine, leucine, proline, and valine, and produces the following fragments when hydrolyzed with acid: proline, leucine, glycine, arginine, proline, and glycine, isoleucine, valine. So in other words, here what we've got is a peptide that has these 6 amino acids in it. We took that, treated it with acid, and broke it apart into a couple of different fragments, and these are the 3 fragments. Can you figure out what the original order of the entire 6-membered long peptide was? I'll let you figure that out. You can pause now if you want, because of course I'm going to give you the answer momentarily. So let's now look at these fragments. You'll notice that these two fragments, proline, leucine, and glycine, as well as arginine and proline, both contain proline. Similarly, these two fragments, proline, leucine, glycine, and glycine, isoleucine, valine, both contain glycine. So here's the deal. Because we are told in this problem that our peptide has 6 amino acids, and there are 6 different amino acids to choose from, it means that we couldn't have used any of these twice. So what does that mean? What it means is that all I have to do is line these things up. I'll start with arginine proline and line it up with this fragment, proline, leucine, glycine, and then line this fragment up with glycine, isoleucine, valine. The original peptide then is this one. So once again, what that means, here was the original peptide. I heated it with acid. Some of that peptide got broken apart to generate arginine proline fragments. Some other samples of that peptide in solution got broken apart to generate proline, leucine, glycine fragments. And some other samples of that peptide broke apart to form glycine, isoleucine, valine fragments. I hope that makes sense. If not, you're welcome to pause it, think about it, and ask me more in class. Now to another question. What fragments would the peptide from the previous slide give if it were treated with trypsin and with chymotrypsin? I'm going to give you the answer, of course, but if you want, you can pause it and think about this for a moment. So here's our answer. I want you to remember from 2 slides ago that trypsin cleaves to the right of lysine and to the right of arginine. So here's our peptide from the previous example. What trypsin will do is it will come down here and find anywhere that there's an arginine, and it will break the bond to the right. And anywhere there's a lysine, it will break the bond to the right. As we scroll down here, we can see there are no lysines. There is an arginine, however. So trypsin will clip this bond. What fragments does that make? It makes one fragment, arginine, and then this other huge fragment, proline, leucine, glycine, isoleucine, and valine. Does that make sense? Let's see if we can answer the next question for chymotrypsin. Alright, we should remember once again from our previous slide that chymotrypsin cleaves peptides to the right of phenylalanine, tyrosine, and tryptophan. So for our peptide in question, we scan down here and try to find phenylalanine, tyrosine, or tryptophan. We scan and scan and scan and scan, and you can see that none of these amino acids are found in this peptide. Hence, well, chymotrypsin is not going to cleave this thing anywhere. So the final fragment that we'd get is actually the exact same peptide that we started with. I hope that makes sense. So I just wanted to show you a computer-generated model of a protein. Now, when I first saw this kind of model back when I was a young undergraduate, I couldn't help but ask myself questions like, what are these purple coils and these green arrows and these twisty green tube-looking things? What in the world is all this stuff? Well, as it turns out, each one of these colored components represents a different part of the protein's secondary structure. Jumping back to our zoomed-out geography analogy from before, a protein's secondary structure is a slightly zoomed-out way of describing or seeing a protein. Simply defined, a protein's secondary structure is the way in which its various peptide segments orient into a regular pattern. The 3 categories of structural patterns are the alpha helix, beta-pleated sheet, and the coil or loop conformation. I'm going to introduce you to these right now. An alpha helix looks kind of like this. So you see this twisty green structure here. This twisty green structure represents hydrogen bonds that are being formed by a twisting backbone of amino acids in our peptide. So this is essentially our peptide wrapped around where there are hydrogen bonds stabilizing it in the middle. This is a sideways view of it. So once again, this twisty green section is a peptide that's twisting into a coil, and that is a type of secondary structure that's often seen in proteins. They're stabilized by hydrogen bonds. The next type of secondary structure is a beta-pleated sheet. Now you can see that what happens is if we have our peptide with its amino acids looking like this, and another section of our peptide that might have been folded over somewhere over here and then is wrapping back, and they hydrogen bond in this kind of manner, they form this type of beta-pleated sheet, which is called a parallel beta-pleated sheet, because the N termini are found on the same end, and the C termini are found on the same end. You can also imagine having an anti-parallel beta-pleated sheet, which would be the converse of direction. I really don't and will not require you guys to know any of these rigid details regarding secondary structure. I just want to make sure that I introduce them to you. Now, I just want to point out one more thing. As we look at this protein structure I was showing you before, you see these little coils? These coils represent individual amino acids in a sequence that are hydrogen bonding in such a way that they coil. These arrows represent parallel or anti-parallel beta-pleated sheets that are once again regions of amino acids here in the peptide that are forming those complementary hydrogen bonds. And then what about all these tubes that are kind of wrapped around all funny? What are those? Well, those regions are just given the name coil or loop conformations. So anything that's sort of an intermediary region between an alpha helix or a beta-pleated sheet that doesn't really fit into one of those two categories, we just arbitrarily call a coil or loop conformation. So that is secondary structure. Once again, if you took a secondary structure and zoomed in on it, you would see individual amino acids, which are the primary structure of the peptide. You zoom out, you see a coil, or a beta sheet, or a loop conformation, or an alpha helix. Those are secondary structural features. As you zoom out more and see this entire type of thing, that's called the tertiary structure. So once again, I would call each of these individual components, beta sheets, alpha coil, or alpha helices, or coils or loop conformations, secondary structures. This entire thing all clustered together in one beautiful picture of a protein is called the protein's tertiary structure. Let me expound on that right here. Tertiary structure really is a zoomed out version that has coils, alpha helices, and beta sheets all being shown here. And if we once again were able to zoom in more closely, we would see the individual amino acids that make these coils, helices, and sheets a reality. In essence, once again, we could say that this zoomed out picture is the tertiary structure of this protein. A couple of details about tertiary structure. The stabilizing interactions that occur to enable coils and helices and sheets to obtain the rigidity or conformations that they have are centered on the presence of intermolecular interactions, which include covalent bonds, hydrogen bonds, electrostatic interactions like London forces, van der Waals forces, and hydrophobic interactions. Now when I say covalent bonds here, there is a qualifier. The only type of covalent bond that exists across a peptide, from one part of a peptide into another part, is a disulfide bridge that we talked about before. So that summarizes tertiary structure. Now what about quaternary structure? If I just told you that we zoom out and see the entire protein and that's its tertiary structure, then what in the world is quaternary structure? Well, as it turns out, there are some proteins that actually have multiple parts that each individually are tertiary structures. And all of those combine together to form a full humongous protein. That is a protein that has quaternary structure. And turns out not all proteins have quaternary structure. The only proteins that have quaternary structure are proteins that have more than one polypeptide chain. So if you have a protein that really is only one polypeptide chain twisting around in one big long pile of coils and loops and helices, that cannot possibly have quaternary structure. But if you have a protein that has multiple polypeptide chains, all individually forming different sections of the protein, then you can say that type of protein has a quaternary structure. Each of those sections could individually be considered a tertiary structure description of the protein. And then the protein as a whole would be called, or zoomed out to see all of those together, would be a way of describing the protein at a quaternary level. As it turns out, proteins that have more than one polypeptide chain are called oligomers. And their individual chains are called subunits. So I could look at one big cluster and say that's an alpha subunit and this is a beta subunit, and they interact together to form this protein. This protein that I've shown here is hemoglobin. Now, although you can't see it clearly in this tangled looking figure, most varieties of hemoglobin have 4 separate polypeptide subunits. Thus, we could say that hemoglobin has a quaternary structure. Once again, any proteins that only have one polypeptide chain do not have quaternary structure. I hope that makes sense. If you want to ask me more questions about it, you're welcome to do so during class. I kind of like to think of quaternary structure sort of as being like, well, when I was a kid, I used to watch Transformers cartoons, and there was a character named Devastator who was made up of like 6 different guys that all combined together to form one huge guy. Each of those individual guys was an individual guy by himself, but when they got together, they formed a huge robot. Quaternary structure is kind of like that. There's sort of like individual proteins almost, but they don't fully function until they get together and form this huge protein like this one, hemoglobin. Anyway, hopefully that tangent was useful for those of you who like Transformers references. Now that brings us to the end of Chapter 23. Finally! And I've got to tell you, I'm exhausted. This was a long one. I look forward to talking to you guys more about this in class, but for now I'm going to turn my attention away from amino acid and peptide chemistry and towards something that might be a little bit more or a little bit less mentally demanding, like researching or looking at my monthly budget reports. So until next time, I'll see you guys later.